Welcome everyone. My name is Erkay Savaş. I am the Dean of Faculty of Engineering and Natural Sciences of Sabancı University. And today I'm extremely pleased to start our fall 2020 seminar series on applications of engineering and medicine and biology. We will have 6 seminars in total that are scheduled this term. All the presenters are leading scientists and well-known experts in their fields. The seminars will provide a recent overview on the most innovative applications of basic science and engineering in research areas such as regenerative medicine, implants, cancer diagnosis, and immunology. You can check the seminar program in our web pages and through social media. Today, our first presenter is Dr. Başak Uygun.
Hello, thank you. If you can let me share my screen, I can start.
Yeah, while you are sharing, maybe I can, say a couple of words about your background.
Okay.
Welcome, Başak Uygun, Dr. Başak Uygun. Dr. Uygun's research focuses on the applications of engineering tools to help patients suffering from organ failure. Her work combines tissue engineering principles with regenerative medicine to develop technologies in the fields of organ reengineering, biopreservation, burns trauma, and wound healing. Dr. Uygun received her BS and MS degrees in chemical engineering from Boğaziçi University in Istanbul and her PhD degree in chemical engineering from Wayne State University. She joined the center as a postdoctoral fellow in 2006, became an instructor in 2011, and was promoted to assistant professor of surgery at Harvard Medical School in 2017. She is the recipient of multiple awards, including NIH Pathway to Independence Award, Mass General Claflin Distinguished Award, Young Investigator Award from American Society of Transplant Surgeons, among others. Her work is supported by grants from NIH and Shriners Hospitals for Children. Her publication appeared in Nature Medicine, Biomaterial Scientific Reports, and Tissue Engineering, and have been cited close to 2,500 times. All right, I'm really pleased to welcome Dr. Başak Uygun. I'm going to turn the mic to Başak Uygun. Thank you very much.
Well, thank you so much for that kind introduction. And I'm extremely pleased to be the first speaker of this exciting new seminar series about applications of engineering in medicine and biology. So it will be given by a group of engineers who are doing incredible work in improving healthcare and other biological problems. So today, as I said, I'm extremely pleased to be presenting my work at the Center for Engineering in Medicine and Surgery at Mass General Hospital, and on trying to, engineering of organ substitutes for transplantation. So this work has been done in the past 10 years, and I'm trying to give you an overview of our accomplishments. So why are we bothering with building body parts? So as you may be aware, end-stage organ failure is one of the leading causes of death in the US and worldwide. And orthotopic transplantation is the ultimate solution for this terrible disease. But orthotopic transplantation has become a very successful procedure, with the survival rates has increased dramatically with the recent advancements in immunosuppressive therapy and surgical techniques. But it's also a disadvantage. The success is also its disadvantage because there aren't enough organs to transplant for the patients that are needing them. So the severe shortage of donor organs are represented by these significant numbers, such that in the US, every 10 minutes, there's a patient added to the waiting list. And unfortunately, on average, every day, 21 people die while waiting for a transplant. So very few of the, not all the patients needing a transplant are getting them because there aren't enough organs available. So there's about 20,000 gap per year. So this gap will become more significant and severe with the increasing incidence of obesity and the advanced donor age over the years and in the future. So there needs to be some alternative tissue or substitute for these donor organs, ways to expand the donor pool or find some alternatives for donor organs. So as some of you might be aware, tissue engineering is promised to do that, right? So for those of you who are not familiar, tissue engineering aims to collect the cells or gather the cells of a tissue or organ in question and place them in a three-dimensional environment called the scaffold, made up of polymeric materials, synthetic or biological, basically like a sponge. So add the cells into the spongy material and culture them or grow them in the lab using a bioreactor system by providing the biochemical and the mechanical stimuli and expect the construct to function as the tissue and organ in question. So this approach is not new. It's almost been 30 years since its first conception by Bob Langer and Jay Bacanti. However, the clinical translation of a tissue engineered product has been very limited. It's the only clinical success is for skin products, like almost the two-dimensional thin layers of tissue that can be transplanted and save patients needing a skin transplant. So it's been very slow. The progress has been very slow, especially for organs that are big and require high volumes and number of cells. So why is it so difficult, right? Remember the cycle, they only need cells and a sponge and the bioreactor, so they're all available. But you need to consider a lot of different points while making this happen. So the first thing that you need to consider is the scaffold, right? The scaffold, how is it going to look like? What kind of material are you going to use? Is it going to be synthetic or biological? It has to have the composition such that the cells will like it and then provide the stimuli so that the cells proliferate, stay viable, and function as you want them to. So it is important to select something that won't cause any immunogenicity once transplanted as well. And the architecture of the scaffold is important. You need to have the right size, right shape so that it fits in its natural position. And it has to have a vasculature for an organ such as the liver, which is highly perfused with blood, that there is an embedded vascular bed in this scaffold so that the cells seated inside the scaffold receive the nutrients that they need. And the waste products are efficiently removed once transplanted. Final and maybe one of the most important things is the cells. Like, what kind of cells are you going to put in the scaffold? So are you going to just put the cells that are responsible for the main function of the liver? Are you going to put the helper cells? Where are you going to get those cells from? Are you going to use human cells or cells from a xenogenic source like pig cells, or cell lines that are produced from cancerous tissue? And of course, if you're creating an organ or a tissue that would replace a human organ or a tissue, you need to have a large volume of cells. So you need to be able to create large numbers to fill in that volume. So all these considerations make the progress in tissue engineering very slow with the traditional methods that it had offered at the time. But about 10 years ago, we and others have come up with this whole liver engineering approach. So in this approach, what we are proposing to do is to get the liver, the organ, in this case the liver, that has been harvested as a donor liver for transplantation, but rejected for any number of reasons, like low quality, not viable enough. So it's going to be discarded. So instead of discarding it, we collect it and then treat it with a series of detergent solutions to remove the cellular components of this organ. So detergent just dissolves the fatty cell membrane, washes the cells away from the organ, and what is left is this white extracellular matrix scaffold that has the protein structure of the organ. So but it doesn't lose its shape, so its architecture is preserved. What you can do is later repopulate the scaffold with some liver cells from a source that you pick, and repopulate them, let the cells engraft inside the scaffold in a bioreactor culture. So you can use potentially patient-specific cells made from iPS, induced pluripotent stem cells. So for those who don't know, iPS is reprogrammed cells from a patient biopsy, like from skin cells. They reprogram it to a pluripotent state, and then you can induce those cells to create the cells type you want. So you can do it for liver cells. And potentially you will have a liver, a donor substitute that's of patient's own cells. And then, of course, you can transplant it back as an alternative. So this has many advantages, this approach. I've mentioned a couple of them. So the most important, in my opinion, is that the process, the scaffold itself is preserved, right? The architecture, the growth shape is preserved, the vascular bed is preserved. So it is easier to perfuse it with whatever nutrient you want. It can be perfused with blood and can be anastomosed once transplanted, just like a donor liver would. So what we did was, the first thing we did was to show the feasibility before, of course, doing anything human. So we worked with rat livers. We got a rat liver cannulated at its portal vein and slowly perfused it with detergents, a series of detergents. In this case, we use sodium lauryl sulfate, which is basically soap. Over time, very gently, and remove the cells from this organ, and this is evident by this drastic color change from, after the blood is washed, you remained with this brown yellow color, and then over time you're removing the brownness, yellowness, and that leaves you this white, almost transparent extracellular scaffold, decellularized liver scaffold that has the exact shape of your starting organ. The first thing we checked was if the vasculature is preserved, and as you can see from this close-up picture, the stained blood vessels are preserved up to the edge of the scaffold to very fine capillaries. And not only the portal vasculature is preserved, the venous vasculature is also preserved, shown in blue in this corrosion cast image. So this was very exciting. We were able to, show that this actually works, because when we first started, I didn't think liver was known for its ECM strength. So once the cells are removed, I thought it would just dissolve. But this was very exciting to see at the time. So the next thing we did was to reintroduce some healthy cells into the scaffold through perfusion, again, through the portal vein. And as you can see here in this image, the cells are back in the scaffold, although it's not as dense as the starting cell cellular density. And then we were able to put this structure in this perfusion chamber and then cultured it over time up to 10 days, during which we observed its function. And then we showed that these grafts were functional in vitro. So albumin, urea, and total bile acid secretion are three major hepatic functions that we measure that are secreted in the media. And our resellerized liver grafts showed similar levels of production as compared to our gold standard static sandwich culture. So this was a good sign the cells were staying alive because hepatocytes are very highly metabolically active cells. You need to constantly supply them with oxygen. Any degree of hypoxia, they'll just die. But we were able to keep them in high numbers viable during this culture period. So once we were satisfied with this hepatocyte, so the liver cell repopulation, we wanted to line the vessels in the scaffolds with endothelial cells. So this is needed because once you're perfusing it with blood, you don't want blood to see the ECM itself because it will create coagulation. So in most of the cases, as you can see, we see a thin line of endothelial cell lining up the vessels, like here, here, similar to a normal liver sinusoid. Although this was, this wasn't very nice to see, it wasn't all around the scaffold, only about 70% of the vessels were lined with endothelial cells. But in any case, we wanted to show that you can actually transplant them and it can be perfused with blood. So we tried heterotopic transplantation of these grafts, which means that we kept the native liver in place, created some space in the animal's abdominal cavity by removing one of the kidneys and placing the graft in its place. So with this method, when the cells are there or not there, blood was able to fill in our graft and perfuse for some time. Unfortunately, because of our not so perfect endothelialization, we had problems with these methods, and then with the transplant long-term survival of the recipients, which was only for 8 hours. And this was due to the inadequate endothelialization. So the blood clotting was a major problem during in vivo blood perfusion. And this is a picture of a endothelialized and hepatocyte repopulated graft in ex vivo blood perfusion, and you can see how it clots. So what happens is when blood hits an ECM that doesn't have the endothelial cell covering, just clots because it thinks it's a wound. What we did with our recipients was to treat the animals with systemic anticoagulation treatment, but that led to hemorrhage. So the recipients didn't survive beyond 8 hours. So that was the first challenge we tackled after our initial success with the preliminary results. And we wanted to create an antithrombogenic scaffold. So we would do the endothelial cell seeding, but we knew it was not going to be perfect, but what we wanted to do is introduce some anticoagulant within the scaffold so that there is, so that the ECM is not exposed to blood. So for that, what we used was heparin deposition by layer assembly method. What we did was we used a positively charged electrolyte like polymer PDA-DMAC and heparin, and treated our scaffold sequentially with positively and negatively charged to create these bilayers over the scaffold. So that would create a reservoir of heparin on the scaffold itself. And as you can see in these images, heparinized grafts were actually heparinized. The heparin was present. So in the ex vivo blood perfusion experiment, the heparinized grafts performed better than unmodified, some conditions better than others. In the 8 bilayer heparin coated scaffolds, when there are no cells, no endothelial cells, there was minimal to no blood clotting. So this gave us the push to try to put the cells back in and then transplant them and see what the recipients would do. And similar to the initial technique I mentioned after nephrectomy, we transplanted heparinized and hepatocyte repopulated grafts. And so that compared to unmodified scaffolds, the 4 bilayer and 8 bilayer have increasingly less blood coagulation. Unfortunately, all these grafts eventually clotted, wasn't enough. So the recipients only survived up to 24 hours. So we're not there yet, and this wasn't enough. So then we went back to the initial part where we were looking at the endothelialization. So how can we improve that? How can we make sure we're close to 100% endothelial cell coverage? So for that, we used a peptide that was engineered in the lab, the elastin-like peptide with a REDV, a short peptide sequence fused to it. This peptide sequence is taken from the CS5 domain of fibronectin, which has been shown to improve endothelial cell attachment. So this fusion protein was generated, made in the lab, and then we used this to chemically modify our scaffolds so that REDV sequence is presented to the cells once they're seeded. So once we did that, in control livers, endothelial cells were still there, but they were peeling off after a few days. When there was REDV, ELP present, this layer of endothelial cells was more stable and staying there at the time, over time. And looking at the endothelial cell functional markers by RT-PCR, the nitric oxide synthase, vascular endothelial growth factor, and VE cadherin, the REDV, ELP livers had more of these expression levels. And as the final test of whether this actually works, you know, endothelial cells being there works, we did blood perfusion experiments. And then what I'm showing you here is the platelet adhesion, platelet coagulation on these livers. So in red, it's the protein that's expressed on the platelets when they stick for coagulation, and control livers, as you can see, it's abundantly present, whereas in the modified livers, there was very little. So this was really good. We were happy with these results, but there was another challenge that remained. We're not modifying the livers just so they're in the vessels, right? We're putting this REDV, ELP protein everywhere. So once we add the endothelial cells, they go stick everywhere, but we just want them on the vessels. So that was the challenge that Ruben actually took on, trying to restrict the endothelial cell seeding in the vasculature. So selectively activate the vessels, or just seed the cells in the vessels so that they stay there, don't go into the parenchymal space. And as you can see, this picture shows a wide view of a scaffold where endothelial cells are only in the vessels. Very few of them went into the parenchyma. It's not perfect, but we're getting there. So we're very hopeful in watching what Ruben has to do with this project. So the second challenge we tackled was repopulation of the biliary tree in these scaffolds. So one of the functions of the liver is to produce bile. Bile is produced by hepatocytes. The bile acid, if it gets into the blood circulation, it's toxic. So it has to be taken in a separate pathway away from the liver. And that's the role of the biliary tree in these organs. And we know from our decellularized corrosion casting, our scaffolds, in addition to the vessels, the biliary tree is also preserved, as you can see here in the green color. So what Ibn Chen did was to get some primary biliary epithelial cells or cholangiocytes and seeded them into the bile ducts of these scaffolds. As you can see here, this is a bile duct cross-section. You can see the cells are highly populated in there near a hepatocyte island. And these are still proliferating, so they're alive, viable. And they're expressing, they're in close proximity to these hepatocyte islands. And she also showed that compared to a two-dimensional culture, these biliary epithelial cells are expressing similar or higher levels of bile functional proteins. So this was, she also showed that there is no difference in hepatocyte functions once the cells, biliary epithelial cells are also there. So this was very nice, and it was the first report that showed the repopulation of the biliary tree. But what was missing in these images and in this project was that there was no integration of the bile canaliculi that is formed between the hepatocytes with the bile ducts, so that the bile produced by the hepatocytes can be effectively transported back into the bile ducts. So to solve that problem, an MD PhD student from Egypt, EHAB, took on this challenge took on a different approach. So rather than building from top down, we started taking a grounds up approach by creating these little liver microtissues with the biliary ductils. So what he did was he mixed hepatocytes with cholangiocytes and sometimes stromal cells into the mix, cultured them in a non-adhesive plate so that these balls of microtissues form over time. And then he observed what ultrastructural characteristics these have. So you can see here in these images, the phase contrast and the scanning electron microscopy images. When there are cholangiocytes present in this mixture, they tend to populate the outside of the spheroid, and they form these cysts and sometimes these tubular buds that have open channel-like structures. And indeed, when you looked at them more in detail through histology, these spheroids had these open channels within the core, and these were lined with the biliary epithelial cells, as you can see in red here. So what we did next was, this is happening. So the cholangiocytes are trying to grow out and connect to other tubules. So what EHAB did was he collected these microtissues and grew them or incubated them in close proximity with the expectation that they will start interacting and forming some junctions, as shown here, maybe channel-like structures. Indeed, that's what he saw. Here I'm showing you a cluster image. As you can see, the blue shows the nuclei of the cells that are forming, and the green color shows a bile analog dye. So this dye is taken up by the hepatocytes, excreted into the intercellular space, and then it's taken up by the bile duct epithelial cells and taken away. So in a real liver, that's the pathway it shows. So 10 minutes, you can see this the dye is being excreted intercellularly, and over time you can detect some outside presence of this dye, which means they're being excreted into the media. And indeed, when you look at them more closely, we saw these two spheroids that is forming a channel-like structure, as I've shown you here. And it also has this green fluorescence, this bile duct analog dye. So this was very exciting because it's the first time to show ultrastructurally that these kinds of integration can be formed in vitro, in microtissues. So what we're planning to do next is to use these microtissues to repopulate our scaffolds so that there is that integration already embedded in the hepatocytes and bile duct epithelial cells, and then let them grow into their natural pathway in the whole liver scaffolds. So the third challenge we tackled was the scale up, right? So we're trying to solve this organ shortage problem. So we can't do it with rat livers, so we have to do a big scale, also scale up. And of course, as an engineer, we know Scaleup, it's not as easy as it sounds, and several other challenges come about, and that's what actually happened. So initially we were successful, so we just applied the same protocol we did for rat livers, and we did this eventual or gradual change in the color. It wasn't as perfect as the rat livers. But looking at the histology images, the decellularized liver had no cells left, so we were able to remove all the cells from the scaffold. And the vasculature, this is a corrosion cast image of arterial and venous vessels, the vascular bed, and they were both present in our decellularized scaffolds. We also did the resellerization. It's a short and a very quick experiment, but instead, to limit the numbers of cells we used, what we did was we took a wedge of human liver, the perfusible wedge, we decellularized it and then introduced the cells back in. So this was the first time we realized that what we're doing with rats is not going to work with humans, because in the rats, when we perfuse the cells through the portal vein or the vasculature, they just get distributed throughout the scaffold. Whereas in human livers, the cells would just get stuck in the vasculature with the portal vein perfusion. So we had to take a more tedious approach of injecting these cells throughout the parenchyma, so that the cells are in the parenchymal space and the vessels remain open for perfusing the media. So in this short and quick experiment, we found that the resellerized grafts produce detectable levels of urea and albumin, but these are very low numbers, so there's a lot of room for improvement in this resellerization part of the project. But before we went more in depth in there, we're not selecting what we get, right, for human livers for this engineering purposes. We're just getting what is being discarded. So they vary very widely in quality, and we were getting a lot of fatty livers. So this is a picture of a liver that has been through the decellularization protocol for I think 2 weeks, and there was no change in the color during this period. And I was convinced that it was blocked, it wasn't getting perfused, and it wasn't working. But when we did the histology, we saw that there were no cells left there. The yellow color came from the fat. So this was a very fatty liver at the beginning, and we couldn't remove the fat with the detergent, it gets stuck. So this red stain shows the fat deposition. So we had to come up with a way to remove this fat, and this time we just washed it with n-propanol, and that was enough to remove the larger fat particles from you can see there's the change in the color, but it wasn't perfect. You can see the pinkish stain throughout the section. So then we took a step back and then realized, okay, we're getting all these different livers. They come from different donor characteristics, backgrounds, so they must be different. Like, we have to understand what is different and how it's going to affect our decellularization and resellerization outcomes. So since then, we got a lot of livers. I think I counted 16 in this one, and when we decellularized 9 so far, we saw that when we grouped the age and the BMI, we found that we can group them as young, old, high BMI, low BMI. So what Eileen Ajun did was decellularize them all. So you can see we have young and low BMI, old, low BMI, old, high BMI livers. So these donor livers. And then we started thinking about, okay, what is different in these? We applied the same protocol on these livers to decellularize, but as you can see, they show a lot of variety in the end product. So what she did was she picked the age factor and then looked at these livers more closely. So as you can see, young and low BMI and I shouldn't say old for this age group, it's older age group. You can see in the younger group, Resellerization produces a more transparent ECM. It's more uniform, whereas in the older age group, the resulting ECM is more opaque. And looking at the histology, there were no cells left in there, so it's all coming from the ECM itself, because DNA measurement in the young and old livers also showed that we're removing them very efficiently, about 90%. So there isn't any cells left. The ECM is different. So then next, she looked at the total collagen and the sulfated glycosaminoglycans glycans and found that in the old age group, there are more of these. But okay, there's more of these, less in the young, but what exactly is different, right? So she did the mass spectroscopy analysis to look at the matrisome of the decellularized tissues and found that there are certain components that are unique in the young DLM and unique in the old decellularized liver matrix. And looking at the collagens as expected, there are higher levels of fibrillar collagen in the old group, and same with the ECM glycoproteins and the proteoglycans. With the exception of the collagen 14A1, there's more abundance of it in the younger age group, and it's a non-fibrillar collagen, which is known to be effective in the cellular stress recovery and response. So it makes sense that the younger age group has more of this component. So there is a lot of stuff. There's a lot of information in here to figure out, what exactly is different and why is different. But what she wanted to know was how these differences affect the cell function when you put cells on it. So what she did was she dissolved the ECM scaffold that she made in pepsin digested, so it's a spreadable substrate, and put a very primary human hepatocytes from a very young donor. So these are young cells on young ECM, old ECM, or rat tail collagen, which is our control group. They don't look very different morphologically, but when you look at the functions, see, these are the same cells, but what they're sitting on is different. So if you compare the young old with the collagen, you can see the decellularized liver matrix is much better. And if you look at the young only, it is a lot better than the old ECM. So for albuminuria production, it's better. For proliferation markers, it's a lot higher. And there is another marker that we look at, the cell polarization. That's the formation of the bile canaliculi. If it's a nicely polarized functioning layer of hepatocytes, they should have a continuous line of bile canaliculi around the cells. So as you can see in the young, there is more of those continuous lines of bile canaliculi, whereas on collagen or old ECM, it doesn't exist. So the data I'm showing is from human cells, human ECM, but she also did the same experiments, actually more in detail, with rat ECM and red cells, and we see a similar result. And same with the cytochrome P450 activity. So as the ECM substrate ECMH increases, the CYP activity also decreases. So this is, again, not very surprising, right? I don't have to show you data to say that young is better than old. But what is interesting is, what in the young ECM that causes this effect, right? So that remains to be seen. It involves a major effort to tease out all these differences and understanding what changes. And stay tuned. We'll be reporting about them. So as the final challenge that I'm showing you the cell source problem, right? So where are you going to get cells that are potentially patient-specific, and you can produce in larger numbers, so you can fill up the whole organ. So Maria Jaramillo, a few years ago, she did this experiment where she took a stem cell differentiation protocol that's published in literature, very well cited. And she applied this protocol on cells that are seated on matrigel, which is the traditional or conventional substrate, or on decellularized human liver matrix. And what she showed after like a lot of analysis and experiments, that finally the cells that are grown on the human decellularized liver matrix has higher expression of all these mature hepatocyte markers at the end of the differentiation protocol, at the end of 20 days. So as she grew them a little bit longer, let them mature a little bit longer, she showed that these cells start looking like primary hepatocytes, and they have some activity levels in terms of cytochrome P4, cytochrome 3A4 activity. They're not quite as adult hepatocytes, but they're similar to neonatal cells. So they're still immature, but we think that as we let them grow in culture, or stay in culture longer, they will mature, because they're showing glycogen storage property, and they also form these bile canaliculi around. So as the next step to this is all done in 2-dimensional cultures. Eileen came and she took the iPS cells and put them in these decellularized liver scaffolds and applied the same differentiation protocol, this time in perfusion culture. So we're now allowing the cells to repopulate the graft and letting them differentiate in situ over time. And then we're hoping to see hepatocytes at the end of this protocol. So she did this up to 20-day perfusion culture, and she showed that the cells are within the parenchyma, they proliferate, they're still viable. And at each differentiation step, the definitive endoderm hepatic specification, hepatoblast expansion, and the immature hepatocyte derivation, the cells that are on or in the decellularized liver matrix scaffold express lower levels of pluripotency markers, higher levels of differentiation markers specific for that stage. So this is the definitive endoderm stage. As you can see, they're much better, a lot better than matrigel itself. And this is the next stage, hepatoblast expansion and the immature hepatocyte derivation. So this is, it's the 3-dimensionality and the presence of the liver-specific matrix that is deriving this differentiation a lot more efficiently than just doing it on 2 dimensions or on matrigel. So our results are not unique. Other groups have seen this with other, pancreas differentiation or other types of differentiation cells. So organ-specific ECM has the specific cues for deriving the differentiation of these stem cells. So all I told you so far is involved our work in the liver, but we're applying what we know in the liver to engineer tissues for reconstructive surgery. So we're collaborating with the plastic surgeon at Mass General Hospital, Dr. Curtis Cetrullo, to create decellularized and re-engineered alternatives for vascular composite allografts, or hard to shape, hard to replace tissues. And first off is nipples. So women or patients, cancer survivors going through mastectomy need this. Current methods are not satisfactory and leads to low life quality and low satisfaction. So this is work of Ruben Oganesyan, and he's using this time pig nipples. Showed up with the decellularization, you can get decellularized, nicely shaped scaffolds that you can recellularize and implant for in situ resellerization. Similarly, for reconstructing large skin defects, we're doing skin flap re-engineering by decellularizing these vascular patches of skin. This is the work of Elise Lupon and Aileen Ajun. So this is the work in progress too. And of course, the next stage of skin flaps is to create these large tissues, right? This is making a human face. The human face transplantation is extremely rare because you need fresh tissue that matches perfectly immunologically the patient. If you have a re-engineered alternative potentially repopulated with patient-specific cells, it's a major resource that would improve this procedure widely. So this is work of Elise Lupon. So stay tuned, we'll have more to report on that. So in summary, I hope I was able to tell you that the whole decellularized whole liver scaffolds provide a suitable template for generating transplantable liver grafts. However, several challenges remain. It's not easy, as being said. So reconstruction of the non-parenchymal components, adding the endothelial cells and biliary cells is something we're focusing. Once this is achieved satisfactorily, then long-term functional outcome after transplantation needs to be shown. Of course, I haven't told you anything about matrix remodeling over immune responses to these scaffolds with our preliminary work. And from literature, we know that as long as the cellular component is removed in terms of nucleic acids and others, there is very little, there's no rejection to these materials. Of course, the scale-up is ultimately the goal to create the functional liver grafts. And also I've shown you the stem cells can be utilized as an alternative source of liver cells. I've shown you work with our work with iPS-derived hepatocytes, but we also done some work to create biliary epithelial cells from iPS. And while doing all this, trying to engineer the organ substitute, we can learn a lot of things in terms of aging or regenerative medicine, just using the decellularized liver matrix as a tool. So depending on the donor characteristics, like age, BMI, or gender, you can evaluate the regenerative capacity of the ECM and try to understand what causes what. So as I showed you, young was better than old, but what is in the young that's causing this? So by this, you can identify some maybe regeneration targets, and then use those targets to make an older liver or older organ behave more like a younger organ. And you can use this as a substrate platform to create an in vitro platform for disease and aging studies. And of course, lastly, I've shown that the decellularization is applicable to any perfusable organ or tissue. I've shown you our recent work on the VCA, the vascular composite allografts, but others have shown it in, pancreas, lung, heart, kidneys, and other organs. So with that, I'd like to thank the incredible team of postdocs and students. I've been lucky to work with this talented group of researchers. I've shown you work of Dr. Aileen Ajun, Ruben, Maria, Julie, Ihab, and Ibn, as well as Elise. So special thanks to them. Of course, I haven't done any of this myself, except maybe in the early stages. And I would like to acknowledge the New England Donor Services for supplying the human livers and of course the funding resources. So thank you very much. And I'm happy to take the questions.
All right, thank you very much, Başak Hocam. It was a great and inspiring talk for us. I mean, thank you very much. And we got some questions. Would you like to go over them, please?
Sure. So the first question I see is, did you try any other types of coagulant other than heparin during forming the scaffolds or when perfusing with blood? Do you think that would make a difference? We didn't try anything else. Tell you the truth, heparin wasn't a very good choice because the way we were depositing it on the scaffolds, it was peeling off during perfusion. Maybe it would have made a difference, but we then realized that we have to concentrate our efforts in endothelialization. So we kind of let that work stop there and focus our efforts in endothelialization. Very inspiring, thank you. Liver itself is also quite regenerative. Is it mandatory to do the whole liver? Is it possible to do decellularization and recellularization at a very minimum scale to start and expect for further regeneration? That's a very good question. It's true, the healthy liver will regenerate, after a substantial amount is removed and reform its original volume. So, did I miss? Okay. So if we're, I think, targeting a not so healthy patient with a not so healthy liver, I think it's not. I don't know how it will regenerate by itself. You know, what we create in the lab and putting it back in the recipient. I don't think that would regenerate, because I think what makes it regenerate is all these signals that come from the native organ, like all the resident cells there, the stellate cells, progenitor cells, even biolect epithelial cells are thought to generate these signals to induce the regeneration. But if you're doing a heterotopic transplantation to maybe just compensate for an enzyme deficiency, for example, then it might be a good idea to create hepatectomy of the native liver, recipient's original liver, so that there are those signals given to the transplanted graft, and that may induce the regeneration of our graft. I'm just curious, have you tried seeding cells isolated from all donors to seed on the young and old liver scaffold? That would be, so we did put older donor hepatocytes on young and old. So there was some difference similar to what we've seen or what I've reported. But the difference wasn't that significant. So they were closer in, in the levels that they produce. So that wasn't as pronounced. Is it also possible to do partial decellularization, like different doses or cell type specific cytotoxic reagents if available, since injection seems to be required for human liver and keeping at least endothelial cells may be advantages? This is also a very good question. Yes, it's possible to do partial decellularization. We actually used irreversible electroporation to create these pockets where there are no cells. So you can really induce cell death in a very localized region to remove the cells and maybe inject cells there. But in our case, we can't do it because once we get the liver, it's already dead. The cells are not viable anymore. So that would be a challenge to, revive those cells. So that's why we're doing whole decellularization. Okay, last question. ELPs are known micromolecules widely used as drug carriers, but when trying to overcome your second challenge, improving the reendothelialization of your liver scaffold, what was the main motivation of REDV fused ELP specifically, to increase cell attachment or reduce platelet adhesion? Okay, so this is again a good question. So we're using the elastin-like peptide fusion proteins for wound healing applications in the lab. So we're creating these bioactive molecule fused ELP peptides for wound healing applications. So the postdoc who was working on that project said, we can use this with this peptide sequence and maybe modify the liver scaffolds. So that's how we came up with that idea. But it's true, the ELP itself also have coagulative effects, but we wanted to have this, abundant peptide that we can easily manipulate and use to modify the scaffolds. Okay, one more. This is from Rhonda. Hi, Rhonda. Is there a way to determine if there is a maximum age where the liver works similar to a young liver? How would you define the boundary? That would be a very interesting study. I don't have an answer to that. You know, we have to really repeat all these experiments for all the age groups that we have. But as much as I hate to admit it, it seems like 40 to 50-year-old is kind of old already. So 20, 30-year-old, I think, is where we're reaching our peak in terms of our ECM quality. I think I've answered all the questions that were in the Q&A. Thank you. They were very good questions. Thanks for sharing your insight with me.
All right. Thank you very much, Başak Hocam. It was really, I want to repeat that because it was a really interesting and a very inspiring talk. We received very good questions, and we would like to thank for all the attendees.
Yes.
And, listening to us and also, asking such wonderful questions. Thank you very much for that.
Yeah.
And also, I would like to remind of the attendees. So we are going to continue with our seminar series. The next talk is going to be on October 21st by John Bishop, a distinguished McKnight professor, university professor, director of Institute of Engineering and Medicine, University of Minnesota. And his talk is going to be about nano-warming for regenerative medicine. So it's going to be, I guess, a great talk again.
Yeah.
Well, it was nice to have you here, Başak Hocam. Very nice.
Yeah, thank you so much for having me, and I appreciate your audience and all the things you've done.
All right.
Thank you so much.
Thank you very much. Have a nice day for all that. Thank you very much.
I will be happy to answer any questions if they come later. Just send me an email. I don't know if there's a way to see them. Yeah. Bye-bye.